us. Thank you.
Thank you so much. Thank you, everyone, for having me on today. Very sorry to be late to the call for a little mix-up. I didn't have the link, but now I do, and I'm here, and let's get started. I'm going to share my screen, the usual thing. All right, and right. Okay, can everybody see it in presentation mode?
Yes.
Fantastic. All right, so I'm going to talk about Terra, which is a platform for accessing data, running analysis tools, and collaborating in the cloud, which is developed by a partnership between the Broad Institute, Microsoft, and Verily. My talk is going to cover these main 3 points. Why are we moving biomedical data to the cloud? And what is Terra? How does it fit within this context? I'll cover the core capabilities of the platform itself and illustrate them through use case vignettes of actual research and work that has been done on the platform. And then we'll kind of zoom out and look at how all of this fits within the greater biomedical data ecosystem. As you all know, this is not new. The size of the data sets that we've been generating has grown dramatically over the past decade or so. 2 decades, really, starting from the initial Human Genome Project. We're at the point where there are projects generating or aiming to generate up to 1 million, and in the future, there will probably be multiple millions of human genomes for some of these projects. And it's not just a human genome sequence, it's also a lot of omics data that's being generated around that. And so that's a huge amount of data that presents immense opportunities for learning more about how human bodies work, how disease can develop, and so on. But that comes with some huge challenges as well. And so it's important to think about how do we share large data sets in this context. And the traditional approach has been to bring data to the researchers in the sense that various institutions have their local computing infrastructures that researchers have access to. And so the idea is you put this data up for download, individual researchers or institutions download a copy of the data and work on their own infrastructure. Unfortunately, it's not very efficient. And at the data set sizes that we're talking about, where we're getting into the petabytes of data for some of these data sets, it just becomes completely unfeasible to have everybody downloading and working on their own copy. And so a very important solution that has emerged to this problem is to use a cloud-centric approach where we're putting data sets on cloud infrastructure and bringing the researchers to the data and putting tools and compute and so on in a centralized place. And so great. Cloud is a very promising solution, but that comes with its own challenges as well, because if you look at the kind of naked offering offered by cloud vendors, it's often too complicated for average research teams to tackle on their own. There are questions of data security, of managing compute resources, deploying code and tools and things like that, and collaborating on this kind of infrastructure that are really not trivial. And so there's a need for solutions that are a little bit more pre-packaged and where things work out of the box and where researchers can go and find interfaces and tools and so on that are ready for them to work with, so that researchers themselves won't have to worry about implementing these fairly complicated things. And so it's in that context, to make it very brief, that Terra came to be. So Terra is a platform that aims to address this problem by providing convenient and powerful ways for researchers to access data, to analyze it and collaborate, both within institutions, but also across in collaborations across institutions, across different countries in the world, and so on. And so I'll tell you a little bit about what Terra provides in this section on kind of core functionality. And basically, you can think of Terra as a platform that allows you to access data sets that are deposited in cloud repositories. And there's a number of data sets that are already available and more that are being generated. Some are completely open access, anybody can access them. Some are protected by various access restrictions mechanisms, and authorized researchers can get authenticated and are able to access the data sets. But you never have to make a copy of the data set, it just stays where it is stored, and you have access through various tools to ways to analyze that data. And to simplify, there's kind of two main modalities that are currently in use. On the one hand, there's a system for running automated workflows, which is really useful for running repeated, reproducible workflows on large amounts of data. And there's also capabilities to analyze data interactively in environments that feel very much like local infrastructure, but where it's actually hosted on the cloud, and you have access to tools that are pre-installed, plus the ability to customize those environments and install additional tools as needed. So those are the kind of the two main modalities. And the goal is for researchers to be able to, as I have said, access and analyze data, but also share the data, tools, and code that they need in a way that's fully reproducible and enables them to collaborate across the world. So let's talk a little bit first about workflows. I want to make sure that we're all on the same page on what that means, how that works. I'll talk a little bit about how it works under the hood, and then I'll spend more time on how it plays out in practice in some of these use case vignettes. So if you're not too familiar with this, don't worry too much about the details. The idea here is to show that a workflow is typically described as a piece of code that will describe inputs, that will describe different steps, some work that needs to be done, describes how the steps relate to each other, and what are the outputs that are produced. And here, this is a very simple little workflow that shows if you were to do variant calling distributed across genomic intervals, a simplified version of GATK variant calling. And so in this case, you see you have a workflow, and you want to be able to launch that on potentially a large number of samples and have the work get done automatically. And this is what the Terra workflow system enables you to do. So from a workflow, in this case, a simple kind of a simple implementation, but it can get quite complicated. You have that initial script. You provide, through the Terra user interface, you can provide inputs, typically organized as a table, and you'll have your table of samples with the input files identified. You give that all to the system. And what happens under the hood, and again, you don't need to worry too much about the different components, but just so you get a concrete sense of what happens, there'll be services within Terra that will handle the inputs, look at the script definition, decide, okay, these are the tasks that need to be generated, and then dispatch that to the cloud backend, in this case, the Google Cloud, And for each step in the workflow, a single VM, a virtual machine, will be generated, and the system will orchestrate execution of that command, running on the data, writing out the outputs, and will proceed, kind of process all of the tasks in the workflow, and apply that to many, many samples if necessary, all in a way that's completely automated. Now, that looks a little complicated on the surface. Terra offers interfaces, graphical interfaces that make the process very accessible, even to researchers who do not have a, a sophisticated programming background. You can take a script that has been written, and through this process, select inputs. There's interfaces for managing execution and managing finding your results. And this dramatically simplifies the process of running complex workflows on very large amounts of data. And so let's go through a few use cases just to demonstrate kind of the point of the system, as well as some of the lessons learned by researchers who have used this. One recent paper that's come out was the announcement by the Telomere to Telomere Consortium of the completion of the full T2T reference genome, which is intended to improve the analysis of human genetic variants. And they basically, the part that was done on Terra, part of this study that was done on Terra, included taking the whole genomes from the 1,000 Genomes Project. There's actually 3,202 that were sequenced with short reads, doing the variant calling, redoing the variant calling across all of these with the new reference and demonstrating the advantage. I'm not going to go into the detail of the paper itself, but I want to show you a little bit the kind of work they had to do to make that possible, to make this paper. And the processing happens kind of in two phases. There's one phase of aligning the sequence reads to the reference, and you can see that they go from, there's 2 files per sample, over 3,000 samples, so that's more than 6,000 files. They actually have to separate those out by read groups, which are a subset, way of organizing the data into subsets. There's about 10 per sample, so you're getting up to 32,000 files at this point. Aligning all of those individually, then merging them back into the 3,000 or so files that are per sample. Okay, so that's a huge amount of files and a lot of processing. It actually gets even worse in the variant calling part proper of the paper, where for each sample, they will do distributed variant calling that is parallelized across genomic regions to generate an intermediary file called a GVCF. Again, don't sweat the details. The point here is that because of this necessity of parallelizing execution, you end up with a point there where you have over 75,000 files. Then there's various steps in which we merge back into the 24 per chromosome final files that the project produces. The point here is to show this is a huge amount of files to manage, and traditionally this is a huge effort both in terms of coordinating all the work and just the amount of compute that's necessary to do it. They were able to do the entire work in about a week, which would have taken months or possibly over a year on a traditional infrastructure. One of the lesson they learned was the importance of testing. You do test the full workflow on just a few samples first, check that everything works, and then you can actually scale up and then just process in a highly automated way. So that really illustrates the strength of having a an automated workflow system that is highly scalable and that uses the cloud for execution. Yeah, so here it's really the scaling plus the effect of enabling people who wouldn't traditionally be able to run this kind of work. Anybody can now take their workflows and run on their own samples on the same infrastructure, and it should pretty much work out of the box, which is a huge step forward in terms of enabling others to repeat this kind of work, and potentially at much larger scale. The same workflow was used, the same system was used recently as well for the All of Us genomic data variant calling, where they processed almost 100,000 whole genomes in the same way. And you can imagine how the numbers of files explode with that number of samples. So that's kind of illustrating the value and the function of the workflow system. Okay. Another example of using this workflow system, this is a paper from last year. Panoply is a framework for doing proteogenomics in the context of cancer analysis. And this paper this is more about tool development than a discovery paper. This is a team that wanted to take methods that were developed by the CPTAC consortium, which is a proteogenomics consortium that has done a lot of state-of-the-art developments for this kind of analysis. And so the authors here, Diarmani and collaborators, re-implemented all the flagship methods that were derived over the past decade of CPTAC studies and produced workflows per module and then unified workflows that pretty much out of the box re-implements all of these complex analyses. And the goal here is to put the power of this kind of analysis in just a lot more hands, so that more researchers who don't necessarily have that sophisticated computational background are able to apply these state-of-the-art methods to their own data. And so some of the lessons learned here involved the realization that to empower a wider community of users, they needed to make the methods available in ways that are both flexible, but also push buttons. So they generated several resources that they make available through Terra that includes per module workflows, so that you can compose new pipelines depending on your research objectives, but you can also use the full pipelines out of the box if you just want to redo the analysis that they had done. And there's also tutorials that show the methods in action on some open access data, which is very important for teaching and for making sure that researchers can verify that they're running the methods appropriately. Another use case that kind of shows this system in action, plus the aspect of empowering the community, is this recent paper. Well, there's actually two papers here, one from 2020 and one from late last year, 2021, where the viral genomics team at the Broad worked with collaborators to analyze, in each case, sets of samples from COVID outbreaks, one in Boston and then the one in Provincetown that happened last summer. And in this case, one of our colleagues in the data sciences platform helped re-implement a number of pipelines that were already in use in some form, but they needed to scale up and automate and make all of this, all the processing much more efficient, so that the analysis could turn over very rapidly, because there were some public health implications, policy decisions that needed to be made, and so on. And so they took existing methods and re-implemented them within the workflow framework offered by Terra. And that made it possible to very quickly analyze large amounts of data from pathogen surveillance sources, data from public health labs, and so on. So the primary goal there was to analyze those outbreaks and feed data into the policy decision process, right? But it has since then had kind of a broader impact, which is that since then we've started working with the CDC and a company called Pathogen genomics to work with public health labs across the USA, so not just in New England, but across the USA, to enable them to run this kind of analysis by themselves. So traditionally, public health labs aren't equipped with a lot of bioinformatics tools and background. And so what Pathogen is doing the bulk of the work here, but it's taking these cutting edge pipelines, in some cases modifying them, customizing them, and working with the public health labs to equip them to be able to do this kind of analysis by themselves. And that has a huge effect in terms of enabling public health community to be more autonomous and to really leverage the technological innovations that have been benefiting genomic research for some time now. In each case, for each of these, I've included links, and we can send them out as a list of links if there's interest. But there's a lot more detail about each of these stories and lessons learned and so on if you're interested in the blog posts. In many cases, these are guest blog posts by the researchers who did the work and who talk about their experience and so on. So that kind of covers the workflow system. Now I want to touch a little bit on the interactive analysis capabilities of the platform. So Terra offers a system that allows you to basically spin up a cloud environment that is either a single virtual machine or a cluster in a way that is pretty push button. So there are some standard configurations. You can launch one of these cloud environments, and there's several different versions that come with pre-installed packages. For example, one of the very popular ones is the bioconductor environment, which comes with the core bioconductor packages plus RStudio. And so you can launch that and directly be able to start working with the tools that you would normally expect to work with. You can still add more packages if you want, but the default is set up to be a fairly general purpose. And that really reduces the amount of fiddling that you need to do to be able to start working with data. And you can work interactively, as I said, within RStudio. There's a couple of other applications, including Galaxy, as well as Jupyter Notebooks, which allow you to work interactively with data in the cloud with a minimal amount of setup. And so, as I've mentioned, there's these pre-baked configurations. So there's some basic defaults. There's a few options that are maintained by the community, and you can actually add your own if you want to generate your own default image to use within this system. And an example of this, this is just an example notebook, but this is a Jupyter notebook that shows a tutorial notebook that was made by one of the GATK developers to demonstrate using a particular machine learning framework. And what's really neat is that you can package all the necessary code and then share it, and then anybody can go and use that same image, use the exact same compute environment, and not have to worry about, well, did I install the right dependencies in the right order? You can just go ahead and work with your code right away. One example, a use case using the interactive analysis system to great effect, I think, is this is a very recent paper just from this month. From a lab, this is a collaboration across several labs, and they have been studying the transcriptional, what happens transcriptionally to various cells in the course of human lactation between mother and child and so on. And part of it was to generate a single cell RNA-seq atlas on this topic. And when you look at a lot of single cell papers, this is just one figure from the paper, right? And it's presenting the cell types that are present in human breast milk across various phases of lactation. When you look at a lot of single cell papers, there's some of the analysis is getting fairly standardized. There are some very common things to look at, but then a lot of it is highly customized. There's a lot of analysis. There's a lot of data processing and cleanup that needs to happen, normalizations. There's various steps of visualizations and then choosing what to look at in more detail. And that can be very difficult to reproduce based on just text descriptions in method sections. And so what the authors did here, which I thought was really neat, was that they generated notebooks. So they published, as part of their paper, they published notebooks that reproduce all of the analyses in the paper. So anyone can go and actually work through the exact same analysis, the same way they do it, by using these Jupyter notebooks. They also make the raw data available. And here it's interesting, they also take advantage of a component that's connected to Terra. It's called DUOS, for Data Use and Oversight System. And that's a system I don't have enough time to talk in detail about, but it's meant to facilitate sharing data that is subject to data use restrictions, and to automate the process of matching the consent conditions to the purpose of the research that somebody who wants to access the data declares. And so they're using the DUO system to share the data in a very efficient way. And they're also, they've also published some of the, things like aligned count data and annotations, cell annotations. They've published that on the single cell portal. And the single cell portal is a portal that's built on top of Terra that makes it possible, that provides interactive visualizations for single cell data types. And that makes it very easy for people who, again, do not have a advanced computational background to go and perform classic visualizations and so on within a graphical user interface. And that's all connected. It's all connected under the hood. And so there's a lot of efficiency built into the system. And this, to me, is a great example of taking advantage of the various capabilities in order to not just do the work, but also share the work in a way that's going to be maximally useful and empowering for the rest of the community. Okay, and this actually gives me an excuse to segue into the part where we're zooming out and looking at the greater biomedical ecosystem. Now, Terra is not just an isolated platform. It's not its own island. It's really a hub within a data ecosystem that is developing in the cloud. And here I'm showing just a few examples of components that Terra connects to. things like single cell portal, which I just mentioned, which is also something that's built by Data Sciences Platform, as well as the All of Us Researcher Workbench, which is a portal for investigators who are part of the All of Us project to work with data in each time. These are basically interfaces that are specifically developed for a different audience to make sure that that audience gets exactly the tools that they need to access. There's a large number and growing number of data sets that are connected to Terra. And what I mean by that is there are some data sets that are hosted within Terra itself. There is a data repository functionality that I didn't go into, but it's possible to host data in Terra. But there's also many data sets that are hosted by other organizations on other infrastructure. And what's really exciting here is that there's been a lot of coordination between these cloud-based efforts to make sure that they're interoperable and that you can connect. And Terra is also not the only analysis platform. And so we want to make sure that whatever platform you're using, whatever data set you're interested in, you're going to be able to connect, access that data with the appropriate authorizations as needed, and run analyses on it without having to move the data, without having to copy the data, and crucially, without having to pay for hosting a copy of the data yourself. Because a lot of this is about the economy of not having to pay for multiple copies. And I want to show actually a quick screenshot of what I mean when I say connecting to data across platform. This is an example up here of the NEMO portal, which is the Neuroscience Multi-Omic Data Archive. It's a single cell RNA-seq data sets that's part of the BRAIN initiative. And it's hosted on a completely separate infrastructure. And it has its own portal. You can download file manifests and things like that. But the more interesting thing is that you can select. So there's a search function where you can select subsets of data. Once you've decided what data you're interested in, you can, this is like a shopping cart metaphor. You put data in your cart, and then you can check out and export to Terra. And when you click that export to Terra button, what happens is that under the hood, there's a handoff between the platforms. The NEMO portal will talk to Terra and say, here's the metadata. So data tables that list, all the metadata about this, this data set. These are the locations of files, etc., and pass that to Terra. And once you have that in Terra, you can run workflows on it. You can localize some of it and work on it in your cloud environments. You can basically do all the work that you need to do. And that connection, the fact that that connection is managed from one platform to the other, means that you don't have to start downloading lists of files on one side and uploading to the other and so on. That connection happens automatically, and that's very powerful, because that's the key to being able to combine multiple data sets efficiently within the same platform, again, without moving the data, just having the descriptions of where the data lives and being able to launch joint analyses on that data. A similar system exists for workflow definitions. So the DocStore platform also has connections to multiple analysis platforms, and you can search for a workflow that has been shared with the community and then say, oh, I want to export that to my platform of choice. In some cases, you have multiple choices. In some cases, you don't. But the point is that as this ecosystem continues to expand, we need to make sure that all the different platforms and components are interoperable with each other. And so there's, in the US, there's a number of infrastructure projects that are supported by the NIH. And specifically, some of these were directly involved in. So Terra is part, for example, of the ANVIL project. The ANVIL project is the NHGRI, so that's the National Human Genomes Research Institute. And ANVIL is a project that focuses on assembling components like Terra, DocStore, and Gen3 data commons, plus having environments like the Galaxy and bioconductor environments on Terra are a result of ANVIL work. And the goal is to put together the resources that a human genomics researcher would be able to use seamlessly and efficiently to do the work that they need to do in that space. And there's other projects that are very similar with similar components. Terra is often part of these, like Biodata Catalyst, the NCI's Cancer Research Data Commons, and so on. They often have a mix of the same components, some additional ones that I haven't shown. And because they're all mapping to similar concepts, there's an effort to coordinate the work. It's called the NCPI, for NIH Cloud Platform Interoperability, to make sure that these platforms are staying compatible and maximally usable for researchers. And to finish, something that I really want to emphasize is that all of this, and right now it's very much within the US context, but there are international efforts to make sure that we can do this at a global scale. And one of the most important efforts in this context is the development of open standards. So we need these platforms to be open source, to be modular, to use recognized standards, and to respond to the needs of the community. And for that work, one organization that plays a pivotal role is the Global Alliance for Genomics in Health, which you'll often see abbreviated as GA4GH. And that's an organization that's doing a lot of work in the space of framing policy, developing technical standards, and so on, in a way that enables us to build out this cloud ecosystem in a way that will be maximally usable on a global scale to address these kinds of challenges in research, biomedicine, and so on. There's a few, I'm running out of time, unfortunately, but there's a few examples that I can give you of where this comes into play, specifically in the context of Terra. For example, in this graphic, we show a nice little cloud with various data sets symbolized by their logos. And there are challenges there of how do we effectively search across multiple data sets? Once we've identified data of interest, how do we connect to the data? How do we retrieve the metadata in a way that is automated, like what I've described with the NEMO portal, for example? And how do we combine data sets in a way that enables harmonization and federated analysis? Because that's another part. If all the data sets are described differently at the level of the metadata, it can be a nightmare to actually combine the data into a joint analysis. And so part of the work is also to develop schemas for how to describe metadata that will make increased compatibility and ease of federation, so that we can do some of these kind of cross data set analyses. There's a lot of work also, since we're talking about a lot of data that is protected in some ways, where there's data use restrictions, you have to be an authorized researcher to access them. So there's some work being done at the GA4GH level for developing a way to manage researcher identities, so researcher passports, so that you wouldn't have multiple logins for various platforms and various data sets. You can have one identity that's recognized wherever you go. And there's also work happening at the level of how do you describe tools and workloads in a way that makes them deployable on different data sets and on different platforms. So it's a lot of work that's still in progress. We have a ton of lessons learned. There's some very interesting perspectives. I am out of time, so I am at this point going to thank you all for your attention, and I would love to answer questions if you have any.
Thank you so much, Dr. van der Welle, for this great presentation and the rich presentation. Unfortunately, we don't have enough time, so I hope we can address some of the questions that the audience already wrote on the chat box. So I'm going to start with the first one from Jeff. His question is about, the first one is from Sid Cox. What is the difference between a data repository and a data generator?
That's a great question. So the data generator can be either the funder who pays for generating some amount of data, and or the facility that's doing the data generation, right? I'm not really separating those out, but it's basically the people who are paying for and organizing and implementing, taking the biological samples and turning that into data. And now they have a pile of data that they want to share with a given research community. That's more and more the case, that it's not an individual research lab that generates the data and then analyzes it. There's like these organized efforts to have a data generation effort, like the Human Cell Atlas, for example. Somebody's going to organize, that's one, most of the data sets that are here were produced in that way. But then these data generators want to share the data with many research groups to give them access so they can analyze and try various different ideas, right? Because these are data sets where there's a lot of possible uses. And so you need to share that. And typically the data generators don't necessarily have the expertise or want to worry about maintaining the infrastructure to do that. And so the data repositories are the infrastructure that can be managed by someone else, who basically says, okay, put your data right here, and we will take care of getting this to the research community. Does that make sense?
Yes, I mean, I think we need to move to the next question, but please, if you want to exchange something, other ideas, Mr. Sid Cox, please go ahead. If not, I'll move to the next question from Jeff. So his question is, or comments, for less experienced investigators, how should they engage, get sufficiently education about using Terra so they can do research? Is there a concierge service or navigator to help people who are new to the platform? I'm glad, Jeff, you asked this question, because I have same thing, especially supporting the educational and training working group. We need to support the young investigator and also to get some mentorship program. So definitely I'm going to be in touch with you, Doctor, for this question, to follow up with you on that. So please go ahead, Dr. van der Welle.
Thank you. That is a great question. And the simple answer for right now is the somewhat not super readable URL on this screen, which is the Getting Started webpage, and that will kind of orient you to various resources. But to describe briefly, so Terra is supported there's a user education team that develops a lot of tutorials, documentation, and so on. And most recently, and this is very exciting, they've been working on a MOOC, I forget what that spells out to, but like an online course. So they're calling it Terra University, and there's a series of online courses that are structured for complete beginners to go through and learn everything about the platform in a kind of streamlined educational way. There are otherwise, a lot of documentation, tutorials, videos, and so on. There, including quick starts if you wanted to get started very quickly and not go through a whole course. There's different personalities and preferences, and so we try to cater to all of that. Now, once you get started, if you run into trouble, there's a support team. So there's a help desk that you can write to privately, or there's a forum, a public forum, if you want to discuss in a more open setting. And you can use either the team responds very quickly, is very responsive, and will help you, whether it's finding resources, troubleshooting something, or clarifying something that's unclear. So I would say terra.bio, and from there, hopefully you'll find what you need, but don't hesitate to reach out if you run into any trouble.
Thank you. Thank you very much. I think the next comment was giving some information on Terra user. A team is developing an online pub course to help people on board. You can find this. Thank you for these links. The courses are free. Thank you. So yeah, we can follow up on this information. We share it also with our training working group as well. Yeah, thank you very much. Is there any other question from the audience, please? If not, so just to let you know, so our next talk, I'm going to put it on the chat box. Our next presentation is scheduled for May 23rd. The title of this talk is A Journey from Opium Research to the Opioid Cohort Consortium, OPICO. So we hope to see you next time in our presentation. And thank you so much. Sorry, is there any question before I close this meeting? Any other comments or question? Okay, so thank you very much, Dr. van der Weer, for this great presentation, and I'll be in touch with you for your presentation and also to follow up with you in some tutorial materials or any other information that we can provide to our young investigators under our mentorship program. So I'll be in touch with you very soon. So thank you everyone for your participation and for attending this meeting.
Thank you, everyone. Thank you so much, Latifa.
You're welcome.
Thanks, Geraldine.
Thanks.
Okay, thank you again. Bye.